to our Research Connect series. My name is Paresh Misra. I'm a transplant nephrologist, and I recently completed my PhD in the lab of Dr. Christina Nostro, studying the immunogenicity of stem cell-derived islets. I'm very honored to be the CDTRP trainee that will be facilitating today's webinar. First, I'd like to acknowledge that the CDTRP is hosted by the University of Alberta, which is located on Treaty 6 territory, a traditional gathering place for diverse Indigenous peoples, including Cree, Blackfoot, Métis, Nakota Sioux, Iroquois, Dene, Ojibwe, Saulteaux, Anishinaabe, Inuit, and many other whose histories, languages, and cultures continue to influence our vibrant community. Myself, I'm physically located in Toronto, which for thousands of years has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. This meeting place is still the home to many Indigenous peoples from across Turtle Island, and we are grateful to have the opportunity to work on this land. I encourage you all to write your own individual land acknowledgements in the chat box. We'd also like to acknowledge Étoiles and CIHR, whose generous support enabled us to host these sessions. So before we dive into the main content, I will pass it to Erica for some general CDTRP updates.
Thank you, Brush. Today I have two announcements for you all. The first is a save the date for the PFD Forum. This year's Patient, Family, and Donor Research Forum will be held virtually on June 13th and 14th. Registration is free and it can be done through the link in the chat. The second is an announcement for our annual scientific meeting. This year's annual scientific meeting will be held in a hybrid format with the in-person component in Calgary, Alberta on December 3rd and 4th. We are currently accepting poster and oral abstracts from all community members. And if you're interested, I will provide a link to that in the chat. Thank you.
Before I introduce the speakers, I have a few housekeeping notes. The first is that today's session is recorded and posted on the CDTRP website and YouTube channel. During the presentation, please keep your mics off. If you have a question or comment, you can raise your hand with the raise hand button or type it in the comment section. So I am very pleased to announce our presenter, Dr. Corinne Hoesli. Dr. Hoesli is the head of the Stem Cell Bioprocessing Laboratory at McGill University. He is a biochemical engineer with expertise in bioprocess development, high throughput screening, and stem cell culture optimization. Her research aims to develop bioprocesses to produce and transplant therapeutic cells to treat diabetes and cardiovascular disease. We are all very excited, and Dr. Hoesli, I will hand it over to you now.
All right, I just want to know, do the people in the audience, are they able to raise their hands and that it's visible to me? Okay, all right. So I want to do a quick vote. I just want to kind of have an idea of who among you are researchers, you know, basically like scientists. All right. And then who are more, you know, like patient donors, people, family members. Okay, I just wanted to get an idea of the audience before starting. Okay. So just to restart my presentation, and I hope that this is all visible. Okay. Okay, so today, and unfortunately I'm only with one screen, so I have, you might be seeing the Zoom bar at the top. In any case, today I'd like to present to you the work we've been doing with support from the CDTRP and other organizations on improving the outcomes of pancreatic islet transplants and making them more accessible to people with diabetes. And this is a long-standing collaboration between myself and Stephen Paraskevas here in Montreal, who leads the Quebec islet transplantation program, or I should say the MUHC islet transplantation program. So it's the only place in Quebec where you can get islet transplants for treating type 1 diabetes. And the talk might be a bit technical, but I hope that those of you that are patients, donors, people interested in transplants, you can reach out to me by email, and hopefully I will catch that email and be able to discuss with you separately. So what we're aiming to treat here is type 1 diabetes. And so I will just try to minimize this hide video panel. Okay, all right. So what we're trying to address is the problem of type 1 diabetes, which is an autoimmune disorder where the insulin-producing cells of the pancreas get attacked by the immune system. And a life-saving therapy discovered in Canada is administration of insulin. So that's the hormone that will tell other cells in your body to take up glucose from your blood. And if insulin isn't present, then other cells in the body have trouble getting enough energy to function. And as well as it's basically your entire metabolism is affected, and without insulin therapy, people used to die very shortly after diagnosis. So insulin has been really life-saving, but it's a very powerful hormone. And so that means that if you over-administer insulin, what will happen is that your blood glucose will drop, and that can lead to becoming unconscious, passing out, and even possibly passing away from the low blood glucose availability. And the management of insulin therapy is also quite difficult. So either via injection or via pump, it's very difficult to control the glucose levels in that very fine range that cells in our body are able to accomplish. So it's a daily burden. But as I said, it's been really life-changing for people with diabetes. In 1921, the discovery of and purification of insulin by Banting, Best, and Collip in Canada. And there's been many steps towards actually a transplantation solution. So instead of replacing the missing insulin, you can envision replacing the insulin-producing cells of the pancreas. And so in 2000, another breakthrough from the group in Edmonton was how to actually achieve more robust and consistent regulation of blood glucose levels, sometimes even avoiding the need for insulin altogether for many years through the transplantation of pancreatic islets. So islet transplantation consists in taking the pancreases of donors that don't have any brain activity and harvesting the islets of Langerhans, which are identifiable in sections of the pancreas as little clusters of cells. They kind of really function like a small organ within the organ, so they could be termed an organoid. And there's about 1 million of these in your pancreas, and you can purify them. And that leads to a graft size that is quite small. It's only a few milliliters, 1 or 2 milliliters, up to 3, that need to then be reinfused into a site where they can live and function. And one of the better sites that have been found, even though there's still some limitations to the site, is the liver of the recipient. So what happens is these islets get reinfused into the liver, and they will lodge into the vascular system via administration through the portal vein. And there they will engraft, not all of them. Many of them are lost early in the transplant procedure and in the days following the transplant, but those that survive can get revascularized, and then they will they will take up glucose from the blood and secrete insulin and other blood glucose regulating hormones into the bloodstream. And when this works, it's really quite effective in bringing back blood glucose control into a physiological level. So even somebody that is very, very invested in managing their blood glucose levels as a person with type 1 diabetes, even with the best efforts, and even with artificial pancreas systems that administer insulin and glucagon, it's hard to get as exquisite control as you can see down here with a functioning islet graft. So this is really amazing, and it can really improve the quality of life, because even those patients that need to go back on insulin, their risks of having low blood sugars, life-threatening hypoglycemic episodes, the frequency of those events decreases after these islet transplants. Okay, so why aren't we treating everybody with islet transplants? Well, there's not enough cells for the transplantation, and also the cells, even if we could mass produce them, not everybody would be eligible. In fact, only a small population would be eligible, because currently you need immunosuppression for the rest of your days after the transplant so that it doesn't get rejected. But immunosuppression also comes with risks, such as increased risks of infection and cancer and other complications. So this is not a negligible risk. And when we make decisions in the healthcare sector, it's always by weighing the risks and benefits of an intervention. So if we want to make this accessible to more people, ideally we mass produce these islets from a source ideally other than adult human donors. And we find a way around the need for immunosuppression, or at least reduce the harshness of the immunosuppression. So this kind of schematic taken from this paper illustrates some of the hurdles and anticipated solutions of some of these challenges. So we could manufacture cells from stem cells, for example, or harvest them from pig sources. But we still would be dealing with the problem of survival after the transplant. So can we engineer better transplantation sites that avoid the early losses? For example, through accelerated vascularization and reduced injury and inflammation right after transplant. Could we, for example, circumvent the need for lifelong immunosuppression via creating barriers between the graft and the immune system, or maybe genetically engineering the graft? And then finally, if we're going to use a device for encapsulation and protection of the graft, how can we engineer those materials so that we improve the function of devices where the cells might be encapsulated? And then finally, to be considered, and I don't have, we're just, starting to address some of these issues of cost, because if we make these kind of complex therapies from stem cells, for example, the manufacturing costs and also the implementation costs will be non-negligible. And that will also limit access in a universal healthcare system, such as in Canada, for reimbursement. Okay, so in the talk today, I'll talk mostly about the production of these insulin-producing cells from stem cell sources and how we address this as a group that works as chemical engineers, as well as technologies potentially for encapsulation. So in my lab, I've been very fortunate ever since my PhD to collaborate with Tim Kiefer. And I was also fortunate to spend some time with one of my students in the Kiefer lab to learn how to differentiate pluripotent stem cells into maturing insulin-producing beta cells through this protocol. As you can see, the protocol is quite extensive, and the starting material are pluripotent stem cells. In our case, we're just working with embryonic stem cells for now. We also have induced pluripotent stem cells, but these stem cells, they are embryo-like, so they can make any cell type in the human body, and they can be proliferated theoretically indefinitely to form more stem cells. So you can expand them potentially in bioreactors to make enough cells that you could treat hundreds, thousands of patients possibly, even from a single batch you could envision. But the protocol to differentiate them and bring them all the way to a specialized insulin-producing cell is quite extensive and intricate, because essentially you have to start from a cell that is embryo-like and bring it through the different steps of embryonic development, just like what would happen, well, not just, but trying to mimic what would happen during the fetal development. And then ideally doing this in methods that are scalable. So the methods we currently use is first to bring them to this stage 4 pancreatic endoderm stage in adherent planar culture, and then aggregating the cells, because islets, as I said, are cellular aggregates, and from then on to continue the culture in suspension in different formats of culture. And we can validate that the cells express the different markers, transcription factors that you would expect to see at different stages. And at the end, the cells can be stained with ditisone, which is a dye that binds to zinc, and the zinc is found in insulin granules as an example. Okay, but all right, just going back to the manufacturing challenge. So ideally, we would like to do much of this process in suspension, because when you do things in suspension, you have a good control over pH, over nutrient addition. You can automate a lot of systems. But one thing that we've noticed in our scale-up, or at least in the suspension culture steps, and this has also been reported by the Kiefer Lab, and that is that during those steps from stage 4 to stage 7, you actually have quite substantial losses in cell numbers. So there's a decrease in yield, even though the cells expand significantly up to stage 4. Thereafter, in fact, we observe some cell losses, and that's concomitant with an increase in the diameter of the clusters that are in suspension. And also the clusters become more heterogeneous. So we start with a very uniform population of these clusters, but then some of them merge together and become larger. So the problem with that is that the larger the ball of cells, the more cells in the ball are consuming things like oxygen. And so the oxygen concentration at the surface will be higher than the concentration at the center. And it will be the same for certain factors that diffuse into these cell clusters. And also there will be gradients in molecules that the cells secrete. So all of this can lead to higher heterogeneity of the product that you're producing. And if you're producing a more heterogeneous product, then the characterization becomes more challenging, and it can also be of more concern in terms of the consistency of what you're delivering to the patients. Okay, so we were thinking, what if we actually encapsulated these cells to avoid this kind of agglomeration process? And also, by encapsulating the cells, we can protect these cell clusters from the mechanical damage that can occur during scale-up in suspension. And I'll illustrate this here. So when we go for scale-up, so when we go from a dose for animal studies to clinical scale doses, we will likely be using stirred suspension systems in bioreactors that have an impeller for the mixing. And that impeller can be horizontal, it can be vertical, there's different impeller designs. But in all of these designs, what happens is that the shear that is experienced by the cells in the impeller region, because this impeller is moving, whereas the container is stationary. So what you get is differences in velocities in the fluid, and those differences in velocities are the highest in these regions in red that you see, and those can impart some damage at the cell surface. So if we encapsulate these stem cell-derived islets in a hydrogel, then they can be protected from the velocity gradients that would occur at the surface. So imagine a bunch of bees in suspension floating around instead of aggregates directly. So how do you encapsulate these cells? You can use conventional encapsulation methods, such as those illustrated here. Most of them are using a nozzle system. So one very common material that you can use to encapsulate the cells is alginate, which is derived from seaweed. And alginate will gel, so it's a polymeric solution at a certain pH and ion concentration, but when it contacts calcium or barium or other divalent cations, it gels together. The polymer interacts with the cations, and it'll create a more gelatin-like substance. And so what you can do is you can have these pancreatic islets mixed with alginate in a barrel and then extrude it through a nozzle. And via different methods, you can get very small droplets that fall into a gelation bath, and then they will form these gel beads, and now you have your encapsulated islets. But these processes are drop by drop, and they can be parallelized, but it's very difficult to obtain a very good distribution between parallelized nozzles. And even when you do, often the droplets that you obtain are teardrop shape. So what we developed during my PhD in the Puree lab in Vancouver was actually an emulsion process. So instead of making the beads drop by drop, we actually mix it in an oil, just like when you make vinaigrette at home. Except here, the calcium is already in the gel, but in a form that is not soluble. But you can trigger its solution and then gelation of the drops as if you kind of harden the emulsion in place. And then you can harvest the encapsulated cells. And actually, quite surprisingly, the cells survived quite well to this process, which can last only between 10, 20 minutes, and you have thousands, millions of these droplets that you can, and this is the kind of process that you can scale up. And you can read up more on this in these different papers down here. But the droplets, as you could see, they're not uniform diameter. So at McGill, we developed another process, which is a bit more like a nozzle process, except it's a emulsification membrane. And more precisely, what we are making is laser micro-machined microchannels through a quite thick Teflon plate. And we can make large numbers of quite uniform droplets and encapsulate the cells this way. So now we get much more uniform droplets. And this can be highly parallelized because it's a membrane type process. Okay, so now we've got our encapsulated cells, and we could see that these stem cell derived islets also survive this process quite well. So you can see they're quite viable within the beads, and they remain viable. And you can prevent this fusion that's happening in stirred suspension. So now you can get much more uniform stem cell derived islets at the end of your process, even after weeks of culture. And you can easily recover these from the beads just by chelating the calcium. And this is just to show the size distribution of the clusters. We get much more uniform, and we maintain, there's no significant difference in the expression of glucagon or C-peptides, which is a surrogate for insulin, in these cell clusters at the end. And they do respond to a stimulus of glucose increase by secreting insulin. Okay, so let's say we can mass produce these stem cell derived islets in a very consistent way. So far, there has been less success when scaling up transplantation methods from mice to larger animals, and particularly humans, and particularly in the field of encapsulation of islets. So one of the reasons that can explain this is that there's a lot of technologies that have been used to encapsulate islets and then transplant them and avoid rejection that can protect the graft from rejection, and they've been quite successful in rodents. But when we move to the human trials, many of them have failed in these kinds of devices for transplantation. One of the reasons can be the difference in how oxygen is provided to the graft when you put the graft into a device for transplantation. So this is an example to the left of a beta cells. So this is insulin-producing cells that are encapsulated in one of those alginate microbeads. And we can clearly see that the cells at the center, when the diameter of the cluster becomes large enough, we can see cell death at the center. And that's because of these gradients in oxygen that I've described earlier. But in the body, our body solves this oxygen distribution problem by very dense vascularization, particularly of the islets in the pancreas. They're one of the most vascularized systems in the body. People have tried to engineer all sorts of different device geometries for protecting the grafts from the immune system: tubes, sheets, pouches, even devices where you have blood circulating through to try to replicate a little bit the vascularization of endogenous tissues. Or another solution is actually to increase the oxygen supply to the graft, but this requires connection to an external oxygen source or some kind of chemical reaction to increase the oxygen level at the surface of the device. So all of these have pros and cons that I'm happy to discuss at length if you're interested. But to study these limitations, what we're developing are ways that we can study this without always using animals as a scale-up step. So we've developed this kind of a device that we can insert tissues. So in this little yellow chamber here in the basket, we can insert a centimeter scale tissue and apply some flow and maintain this under culture for several weeks. And we can also image these tissues through the top via small animal imaging systems. And we can monitor these tissues over time. So instead of using, it's almost the size of, a pretty large tissue that would be in an animal, except that instead of using an animal, we're using this kind of in vitro system to look at larger tissues and how they behave in different oxygen environments. And we can look at how the tissues respond to changes in glucose, as you see in red, by secreting insulin, because when we're using this, we're putting it under flow with a pump. And so we can harvest periodic samples to check for insulin production. Okay, so what this has allowed us to do, and this is under submission right now, is to really look at the effect of different cell quantities and different amounts of oxygen. So at the top here is different concentrations of oxygen, actually partial pressures applied in the flowing medium. So you can imagine the pump that we have in that system is almost like a heart. And then we have this tissue that's being perfused. And in this case, there's some channels through the tissue so that we can look at the effect of different parameters on the survival of the cell and the performance. And we can see definitely that the more cells we load, the less viable cells there is, or the shorter the distance at which the cells are still happy. So that means there's an oxygen gradient. And when we reach a critical threshold, then we get quite abrupt cell death. And when we decrease the oxygen supply in the channel, then that viable radius further decreases. And from this, we can come up with different viability radii and do more engineering to try to figure out the optimal design or minimum criteria for the design of a kind of transplantation device. And what you see down here is numerical modeling to predict the oxygen tension concentrations. And as I said, we can even image this. So by time-lapse imaging from that window that you saw at the top, we can see how long it takes to establish a kind of steady state of survival. So, and we can look at different types of tissues. So we can have different scaffolds where we can include not just the islets, but vascular cell types and look at how the revascularization might be happening in these artificial tissues. This was a collaboration with Paul de Vos in the Netherlands. And with Aspect Biosystems, as well as Tim Kiefer in Vancouver, we also looked at 3D printed fibers, which is another geometry that people are investigating for delivering these stem cell-derived islets. So in this case, we're just using one fiber. So the surface area is very large, so we can get quite high survival in this situation. Okay, But what if we wanted to deliver about 600,000 islets, which is, let's say, a current average of the dose that you need for a therapeutic impact in terms of eliminating the need of insulin exogenous. So what if we try to mimic this vascularization that we see in the body, but still have the cells protected from the immune system? What we're trying to engineer is a device where you would have a hydrogel like alginate, but it could be another hydrogel that would provide immunoprotection. But inside of that gel, we would engineer some vasculature so that we can distribute the oxygen well enough for survival, but still avoid the direct contact between the immune cells and the graft. So one way that you could imagine doing this, these kinds of intricate branched architectures, is via 3D printing. With a team in Quebec City at Université Laval, we have been working on this sugar 3D printer. And this was inspired by a previously published work. So as you can see, this was based on work by Jordan Miller, but we adapted the method so that we could have a 3D printing nozzle with quite well-controlled temperatures. And from this, we can obtain what looks like caramel structures, but my students aren't happy when I use the term caramel because it's not actually caramelized. It's actually a carbohydrate glass, but it's sugars from which we evaporate water and that form polymers that then can create quite well self-sustained materials. Just like when you have a dessert and the chef has a fine hand, is able to draw very intricate caramel structures. It's similar to that, not exactly like that, but also 3D printed. And we can look at the print fidelity in collaboration with Marc-André Fortin's group and also in Quebec City, and create these branched architectures up to 16, maybe even 32 branches, again, kind of hierarchical vasculature. Usually we use a system with one inlet and one outlet. So one of the possible issues with this strategy, so we could use the sugar and we could actually cast cells around and then the sugar can dissolve during the casting. But one potential issue is whether these caramel or not caramel, but polymerized sugars might have an impact on the cells that you're putting around. So Jonathan Brassard developed, so this is a student in my lab, developed a method for coating these sugars with a polymer so that we actually dip coat the sugar networks first with this polymer, and then we dissolve away the sugar. And now what we have is a kind of vascular graft system. So this is very similar to the devices people use for bypass surgeries, except that they have branch channels. And then we create an external compartment so that now between the external compartment and the internal vascular templates, we have space to put the stem cell derived islets in the hydrogel. So now we can fill this and we will have our cells just like in the figure I showed earlier, kind of like this figure, right? So this is the concept. We have these architectures and we'll have the cells around in the hydrogel. But the beauty of this is a off-the-shelf type system. So we could basically prepare this, sterilize it, clean it, and have it ready for infusion of the hydrogel in the cells and transplantation. But of course, there's some optimization to do in terms of the geometries to make sure that the blood distributes well through this. So let's say you were to graft the two extremities to existing blood vessels. How will the flow distribute? Will you get any kind of blood clot formation? How will the oxygen and insulin distribute to diffuse to and from the devices? So again, we're looking at different modeling strategies to investigate that. And this is where we're at. So we're working on these kinds of strategies, different ways of 3D printing, different ways of engineering these devices. Another consideration I would say is, again, the cost and simplicity. So the simpler the process, the less production steps, the more ready-made things can be, the better. And so I'd like to thank everybody in the lab that's been working on this. So we have different research topics in the lab, so not just on diabetes, but on cell therapy in general. And this is a relatively recent photo of the lab members. And I'd like to thank particularly collaborators on the CDTRP for support, as well as various funding agencies. And I'd be happy to take some questions.
Thank you very much, Dr. Hoesli, for that really intriguing presentation. So I'll open the floor to any questions. Let's see if there's any. You can also feel free to put the questions in the chat. In the meantime, Dr. Hoesli, if I may, I have a few questions of my own. I was quite mesmerized by some of the technologies you're developing. You talked a bit about nutrient diffusion, and one of the questions I always wondered with these hydrogels is, if these islets are sitting inside an alginate capsule with a hydrogel inside, does the hydrogel itself impact diffusion in any way?
Yeah.
And is there
just before the talk, I removed slides on that because I thought it was intuitively, I would have thought not that much. I calculated some of the diffusion coefficients during my PhD, and but in actuality, after measuring it, actually can be substantial. So, but however, the impact of the hydrogel depends a lot on how many cells you have and the oxygen demand. So in engineering, we have this, basically it's a problem of reaction diffusion. So if you don't have many cells, then the diffusion can become a kind of a quite, because then the distance is longer before you reach that critical distance, and then it can become more limiting. But if you have a lot of cells, then the amount of cells is more dictating of the behavior of the system. So what we have is this dimensionless number called the Thiel modulus that we often use to compare reaction and diffusion and the different predominance of those different phenomena. So in systems, so to be short, the impact of the diffusion and the hydrogel, it depends on the oxygen demand of your system. So in certain cases, it becomes quite critical, and in other cases, what's more critical is the consumption rate, and the impact of the gel versus water is not as much. Yeah.
So it seems like it's an interplay between like the cellular metabolism and the hydrogel itself.
Yeah, well, it's basically how important is the diffusion parameter, and it's like because water and because when you have just diffusion, water also has a diffusion coefficient, and then the hydrogel will actually lower that diffusion coefficient. But if you have a lot of cells, then the distance alone, the oxygen will drop very fast. But if you have fewer cells, you have a longer diffusion distance, and then the difference in your diffusion coefficient becomes more important. Does that make sense? So the lower the amount of cells, the more the diffusion coefficient is important.
Got it. Okay, so there's competing sort of parameters at play. Yeah. Okay, it's more complex than I'd imagine.
Yeah. However, I can tell you it's significantly lower than just water in most alginate formulations. And I wasn't thinking that because hydrogels are 98% water, but it's still actually the hindered diffusion is actually creating more impact than I thought before measuring.
Got it.
So
Thank
very
you.
long answer to
Yeah.
that question. Sorry.
Something worth thinking about, though. It speaks to the complexity of the systems you're working on. So, very impressive work overall. I think there's also a question from Jonathan Choi in the audience.
Yeah, that was great stuff. So I got a couple questions. I guess the vascularized system that you ended on, have you been able to test like the permeability of that to like antibodies complement immune factors?
Well, the polymer itself that we're using for the coating, it's highly permeable, and we designed it that way because we didn't want that to be the main barrier to diffusion. The hydrogel itself, it depends what you're using as your hydrogel. So you can formulate alginate to largely exclude antibodies, but then the diffusion coefficient drops, of course, for other smaller molecules. But with alginate, it's very difficult to completely exclude antibodies, but you can formulate gels that largely exclude antibodies.
Okay.
But it depends. There's other gels that you can use that would be more immunoprotective. And then the role of antibodies in the mechanisms of islet transplants in an encapsulated context is also debatable. So yeah.
Okay. And then I guess I'm interested in stuff getting out the other way, right? So what's turnover like in these I guess islets that you're grafting in, right? Like how long does a beta cell last when it dies, right? Is it cleared by other beta cells, or do you need like, say macrophages come in, get rid of it, and then you're struggling with the I guess the movement of stuff outside, right?
Yeah, well, physiologically, islets are quite long-lived in humans. Of course, their half-life is something like 4 years or some. It's very long by radio tracing experiments. The stem cell-derived islets, we haven't measured it personally. I believe that when they're mature, they also have quite low turnover, but certainly there might still be some proliferating less mature cells in that cellular aggregate. And then once you encapsulate it, for example, in a hydrogel like that, how long-lived will it be? What I can tell you is that in NOD mice, we don't see a drastic change in the diameter of these clusters after 4 months. So I don't know if that's from death and turnover. But one of the things with the gels is when the cells die, you actually see it; they accumulate, and we actually see it quite well. When we have a graft that doesn't function, we can see like an empty space where the aggregate used to be. We can almost measure the change in diameter over time, like by the size of the gel space versus the cellular aggregates surviving in the space.
Sure. Okay.
That's a great question. Yeah.
Thanks.
I see there's also a comment in the chat from Tetsuya Kin. Great talk, Corinne. We can provide human islets with you for research if you need from the Alberta Islet Distribution
I already
Program.
am getting them. I'm already getting them. Yeah.
I think the whole community is grateful to the Alberta Islet Distribution Program. I don't see any other questions in the chat for now, so maybe I'll take the liberty of asking another one. So I really, I'm always fascinated by microperfusion systems, and I think that they're probably very complex to develop, but obviously if there's expertise, then, I think about some of the research questions one may have. Do you know if it's, first of all, if your system is amenable to co-cultures, like different cell types? For instance, you put your islet in there and then any other cell type of interest that you would be interested in studying interaction.
Yeah, one of the figures I showed there was Min6 beta cells with the HUVEC cells. So you can co-culture it under perfusion like that. And then the only problem is that, at least in our system, sampling is difficult. We can do this imaging, but it's low resolution. Of course, microfluidic systems might have higher resolution, but what we're trying to achieve here is this kind of scale up. Because what's been happening in the field is that people develop very successful strategies in rodents that don't transfer. That's why we're aiming to make these kind of bigger tissues, but it does make it more difficult to monitor over time what's going on. You can have different cell types with different reporters in them, so you can kind of track, but again, it's kind of low resolution imaging. But we can measure things that are secreted from the tissue. But it's still, it's very user-friendly. Anybody that wants to use that system, reach out to me, because we can make one for you and explain how to use it. It's actually, once it's engineered and validated, it's actually relatively simple to use.
Very cool. I'm always interested in the immune system and how they interact with islets and stem cell derived islets, and I wonder if this platform might be. Yeah,
we have an immunologist that hopefully will join soon, and that's what we've been discussing, exactly what you're saying. Like, have your vascularized tissue and then send some different immune cells.
Exactly. Absolutely.
What's happening. Yeah.
Very cool. I see that Erica has her hand.
Yes, very good presentation, very interesting work too. I was wondering if you could talk a little bit on what would be the next steps for this project.
Well, one of the next steps is what was just mentioned about, circulating these immune cells. That certainly would be very exciting. We've been working on kind of hybrid technologies where you could kind of combine 3D printing with reorganization of the tissues. We've also been conducting actual transplants in pigs with these devices to check for safety of the procedure, because that will be a high-risk procedure as well when you're talking about a vascular surgery. We're quite interested in collaborating with groups now that we have like a pretty good setup to test vascularized artificial tissues. We'd love to collaborate with people that bring in other cell types and other types of tissues that we can study. And what personally, my own lab, where I'm interested in the scale-up aspect and the process control, because these are 4 weeks, 6 weeks, 8-week cultures. The more automation we can bring into this, the more process control, the more kind of conventional chemical engineering strategies, quality by design, the more of that we can bring into the process, the better. The only problem is that it's very expensive. So, and the other thing, I don't know if I guess people are here from Edmonton, maybe on the call. Yes, right? Yes. I would love to have a bit more across Canada discussions on bringing down costs and collaborating on the manufacturing of these therapies in Canada, because I don't think we want to wait for foreign companies to have very expensive products. Like, I think we can make this in a coordinated way within Canada and try to bring down have a different business model, perhaps, than a private production model. And I'd like to discuss this with different people across Canada that might be interested.
Definitely a lot of groups across Canada, and I know BC, also Toronto over here locally, we have a few, so always open to collaboration.
Yeah, but I think we really need to sit down together at a table and think about, can we make this in Canada? Can we make it in a way that is less costly and coordinated, and that we address some of the challenges that these quite expensive therapies will bring to the healthcare system? Yeah.
Absolutely. So I see there's a comment in the chat, actually. I would propose the CDTRP annual scientific meeting in December as a venue for that discussion. So I think that's an excellent idea. I don't know, Erica, if there's any way to facilitate that, but
100%. I can easily talk to Corinne after that, and we can see what our membership looks like in diabetes, and see if we can get some type of working group or just the word out.
Yeah, and we've also, I have to discuss with others, but there's also Diabetes Canada meeting. Perhaps that could be a venue, but perhaps that could also be facilitated by the CDTRP. I'm just putting it out there. I don't know if, yes, this part is still being recorded. So anyone that's hearing this, I'd love to chat about this idea. But I'll be reaching out anyways.
Wonderful. A last call out if there's any other questions or comments from the group. If not, I'd like to thank everybody for the questions and comments. And of course, Dr. Hoesli, on behalf of the CDTRP, thank you again for presenting. And if anybody has any other questions, comments, or suggested topics and speakers, please contact Erica Croft at ekcroft@cdtrp.ca. We can put that in the chat. I just want to mention that all participants will receive an email asking them to fill out a survey, and we would appreciate if you took a couple minutes that it takes just to complete it. So thank you, everyone, and enjoy the rest of your day.
Thank you.
Thank you very much. Goodbye.
Thanks.